get started. Okay, so the first thing we're going to kind of look at here is the understanding that mitosis is the division of the nucleus into two genetically identical daughter cells. Okay, so what's going to be happening here is you start off with one cell and you create two new ones. That should be a pretty simple concept for you. And it's a cloning process. In mitosis, we are making two genetically identical daughter cells. In 9th grade, you should have learned about meiosis and mitosis. They're very different processes, right? Mitosis is the creation of identical cells, and meiosis is when you actually get new genetic material entering the cell. And meiosis is this one, right? This is not what we're learning about right now. We're focusing on cell division. Meiosis has nothing to do with cell division. Meiosis is about exchanging genetic material. Okay. So there's 4 reasons why a cell would divide, and these should be kind of obvious. The first reason is to repair tissues or to replace tissues. When cells, get sick because they're infected and they die, you can't just, be throwing away cells. You have to replace them, right? So the first reason to undergo mitosis is to replace damaged, injured, or dead cells. And you can see this, like when you go to bed at night, your skin cells are actually, the outer layer of your skin cells, they're generally dead, right? It's the inner levels of your skin cells that are still living and dividing. These outer layers actually come off when you sleep, and that's why you should be washing your sheet, because you get dead skin cells all over your bed sheets. The second one that we're going to be looking at is the growth of an organism. So if we we obviously all started off as one cell, and now we have millions of cells. And so one of the main purposes of dividing is to grow as an organism from single-celled to multi-celled, right? And just think about those single-celled ones too. They do individually grow as well. Like if we're talking about any single-celled animal cell, they do grow in terms of the length of the cell. The next thing is asexual reproduction. So we're living in the age of the coronavirus, and viruses don't reproduce by their own means. But when we think about other disease-causing agents like bacteria create clones of themselves. Like they reproduce through mitosis. They don't reproduce through meiosis, which is what we do. And so one of the other functions of mitosis is asexual, which is another word it's the scientific word for cloning, right? Sexual reproduction means that you're going to be exchanging DNA. Asexual reproduction means the DNA is going to be unchanged. You're going to form a clone. So this is one of the main functions of mitosis. And then lastly, development of embryos. So when an embryo forms, it's going to, when your sperm meets the egg, right, you're going to be creating a fertilized zygote. And that's going to go through mitosis. So from the first cell to all the other cells, there's no new changes in genes, right? Your chromosomes and the DNA that the chromosomes hold are locked in place. They're unchanging from this point on, unless mutations happen. But mitosis is what takes place from that point to go from one cell to 2 cells to 4 to 8 until you eventually become an embryo. But just because you're undergoing through mitosis, one important thing to understand is mitosis, the thing that's being cloned is the DNA, right? The DNA is being copied. But that doesn't mean that the cell has to remain the same, because it's the DNA, and your DNA is a very long thing. It's the DNA that codes for specific proteins and carries the instructions as to how that cell is going to look. And we're going to learn in genetics that there's different genes within your DNA that can become activated and deactivated. And depending on which genes become activated, you can create an entirely new type of cell. So this is how you get cell differentiation, okay? So zygotes are going to undergo mitosis, and they're going to differentiate to become embryos, which eventually will lead to the formation of organs and tissue systems and entire individuals, right? And your mnemonic here, you know I love my mnemonics. The mnemonic that you're going to be using is TOAD. For tissue repair, organism growth, asexual reproduction, and development. All right, so we finished this mnemonic. Let's continue on here. We are going to look at the process of mitosis. So when we are looking at cell division, we're actually going to be referring to it as a cycle, okay? You'll often hear it referred to as the cell cycle. And the cell cycle, it has basically 4 parts to it, okay? We start off over here in growth and normal metabolic rates. This is the first growth phase and is often referred to as G1, okay? This is the phase where the cell is going to be growing from the point where it was created, right? It's going to be growing. It's doing its normal metabolic things like making proteins, making proteins, making carbohydrates that are needed, creating ATP for use and storage later. And this is called G1. Notice that all 3 of these are interphase. So really, you can split up the cell cycle into 2 main phases: interphase and mitosis. Okay, that's the cell cycle. But interphase has 3 parts to it. So the first part of interphase is what we just talked about. The second part, the S phase, this is when your DNA actually replicates. And when we get into the genetics unit, that's when we're actually going to see, how does DNA replicate? What's the process involved? There's DNA replication, transcription, and translation. And we're going to be learning about those. For now, we're just going to name it. It's replicating DNA. It's making a copy, right? This is your S phase, your synthesis phase. Then the cell cycle is going to continue on into the second growth phase, G2. And this is when we have a copy of the DNA. We're going to do a little bit more growing, and we're also going to do some metabolic things, making some proteins, some enzymes that are going to be used in the cell division process. So this is your second growth phase. And then we get into mitosis. So mitosis is a multi-stage process. You have prophase, metaphase, anaphase, telophase. 4 steps to it. You would have learned about these in Science 9 in the human reproduction unit. And once these 4 phases are done, and don't worry, we are going to review them. I know that was a long time ago for some of you. Once these are done, you have the final phase, cytokinesis. And cytokinesis is a fancy word for the splitting of one cell into 2. So this is when that one cell gets split in half, and by this point, the DNA has moved, separated into 2 sides, and will split into 2 cells. All right. Let's go through the M phase a little bit more. So you have mitosis and cytokinesis, right? Mitosis is when we're going to be doing nuclear division. So our nucleus is going to be splitting, our DNA is going to be splitting in half, right? And we're going to actually create 2 nuclei where there was previously 1. And then, so this is mitosis, right? This is referring to prophase, metaphase, anaphase, and telophase. In these phases, what's happening is our nucleus basically gets ripped in half. And it's not quite so dramatic, right? It's not actually getting torn in half, but one nucleus is going to turn into 2 identical nuclei. In cytokinesis, this last part of mitosis, this is where the contents of the cell are going to be segregated. Segregated is a word that means separated, right? To different areas. There's segregation. And the cell is actually going to split into 2. So each cell is going to have all the necessary components that make it a cell. All right, let's talk about interphase. So remember, interphase is all three of these, right? It's G1, S, and G2. What happens in interphase? Here's your mnemonic, doctor. Okay, this is what happens in interphase. The first thing is the big one, the necessary one, the one hopefully no one forgets, is an interphase. The interphase you can think of preparing for mitosis, right? In interphase, when we're getting ready to divide, the first thing that has to happen is we have to copy our DNA, right? This happens in the S phase. The second thing that happens is we need new organelles, right? If our goal is to create 2 new cells when before there was only one, in order to create a second cell, that second cell would have to have its own organelles, right? Cells, they need endoplasmic reticulum, they need mitochondria, they need ribosomes, they need a Golgi apparatus, they need all that stuff. And so you can't just start splitting apart. You need to make sure that that new cell that you're going to create has all those organelles necessary for it to function. The third thing that has to happen is it has to grow, right? We can't just, if we had a cell, I'm going to draw this instead of trying to use my hands. If we had a cell that was this big, this is a plant cell, it's a square, and we just divided it without letting it grow. Well, then now we have 2 cells, but they're significantly smaller and more squished. Remember, you still have I'm not going to draw the organelles, but you have a bunch of organelles in here and they're squished. If we divide again, oh my God, now the cells are even smaller, right? If we keep dividing without actually growing, then eventually your cells are so small they can't hold the organelles inside of them, right? So let's say that you have a cell that's this big originally with all the organelles inside of it. Before we divide, the cell actually has to elongate and basically become twice as big as it was before, so that when we divide in half, the cells are normal size. So here's cell 1 and here's cell 2, okay? And this was the original cell. So it has to grow before it can divide. So cell growth is an essential function that happens here. And the cytoplasm, we know what that is from the previous unit, that volume has to increase before we divide. So the area that your cytoplasm is in, it has to grow, right? Basically, we're increasing the area of the cell. All right. Fourth thing to happen, transcription and translation. So transcription and translation, we're not going to be discussing what these words mean just yet. This is going to be in the genetics unit, but it has to do with copying DNA, okay? And what we're focusing on in interphase is making the key proteins and the enzymes that you need in order to do this, right? If one of our goals is to create new cells and we're going to be copying that DNA, we need the proteins and the enzymes that actually do that, right? So it's an interphase where we make those key proteins and those enzymes that will replicate our DNA. Next up, obtaining nutrients. Vital cell materials have to be present, right? We know that every cell needs amino acids, it needs glucose, it needs CO2 and H2O. There's vital compounds that are necessary for a cell to live. And before a cell divides, it's going to actually be using a lot of those things, right? So it's almost going to, you can think of it as like hoarding materials, like a chipmunk preparing for winter. It's going to be collecting a whole bunch of glucose and water and CO2 and amino acids and nucleic acids. It's going to be collecting all that stuff because they're going to be necessary in all of these future processes. And then finally, cellular respiration. So we just finished the unit on cellular respiration where we talked about the mitochondria creating ATP. We're going to need a lot of ATP. This is an expensive process. So interphase is the time before dividing where we are going to be making a ton of ATP that we're going to be using in future steps. All right, next understanding. We know what chromosomes are from Science 9. They look like this, right? They look like this. Here's a chromosome. Chromosomes, they actually condense by supercoiling, and this is in mitosis. So in Science 9, you would have learned that, and I think this is the next slide too, that a chromosome is actually going to replicate itself into sister chromatids, and those chromatids are going to separate into daughter chromosomes. We're going to go through this process. So if you forget this stuff, don't worry. But the DNA actually uncoils, right? And we're going to see images of this. And what's important is the difference between chromatin and chromosomes, right? So chromatin, your DNA is loosely packed within the nucleus, and it's basically unraveled, right? I often think it kind of looks like spaghetti in a large pan, right? It's just loose and kind of everywhere. This is the unraveled form. It's very different than chromosomes. Chromosomes are packaged very tightly and densely into these perfect little packages, okay? Chromatin is kind of the opposite of that. It's loose and unstructured, okay? And this is what your DNA is actually like for most of the time. It only really goes into chromosomal form when we're trying to divide. Every other time, it's unraveled and just in the nucleus. This is kind of a hard thing to, like, we know the nucleus is circular, right? And the DNA is just loose in there, kind of like this, right? It's stringy and it's just kind of in there. This is your chromatin, okay? It's still DNA. It's just been unpackaged, unrefined, and it's just sitting there instructing the cell on what needs to be done. Chromosomes are different, okay? Chromosomes are a temporary packaging, and that's kind of the best way to describe it. And what happens is, before you divide, you're going to make these chromosomes. And how does it happen? It's via a process called supercoiling. And it's basically, you take this chromatin that's all loosey-goosey, and you're going to tighten up this chromatin. So here's all the chromatin. You're going to tighten it up, tighten up the coils, and you're going to make these perfect little fibers that are your chromosomes. Okay. And so this happens to make cell division just a little bit easier, right? It's in this form, in the chromosomal form. The reason why we do it is because then the DNA is easily segregated. And you can kind of picture this. Because of the form that it's in, this piece here can go this way, this piece here can go this way. We can split it up, right? Okay, so DNA is in this form in chromosomes during mitosis. And we're going to go through how this happens. Okay, so remember that the DNA is replicated in the S phase, right? So this is a single chromosome, right? And it's going to be replicated. So this is going to be your new piece that gets formed in the S phase. And now we have sister chromatids, right? So these are going to be genetically identical. This new piece here is a genetically identical copy of that original strand. And the name for these are sister chromatids. And they're held together, but you can see this little central region here. This is called a centromere. So these two sister chromatids are being held together by the centromere, and there's your spelling for that word. When these chromatids separate during mitosis, and we're going to go through the different phases, they separate again. So we're going to make a copy in the S phase, and then when this S phase is part of interphase, right? And then when we're undergoing mitosis, and remember there's a whole bunch of different phases, anaphase, telophase, prophase, metaphase. In mitosis is when these two identical strands get split up and go to opposite sides where they become different, identical copies in two new cells. Okay. So we already said mitosis, what we're doing is we're dividing the nucleus in half, right? And in order to do that, we have to duplicate our DNA molecules into two different nuclei. And there's 4 phases: prophase, metaphase, anaphase, and telophase. And those words, they happen in that order, okay? So the division of the cell, which is what we call the splitting of the cell, cytokinesis, it occurs at the same time as telophase. So as telophase is happening, and we're going to talk about what happens in telophase, during telophase, the final phase of mitosis, is when cytokinesis happens. So the reason why I'm kind of saying that is there's often a misconception. No, I don't want a laser pointer. There's often a misconception that it goes prophase into metaphase, into anaphase, into telophase, and then when telophase is done, that's when we split with cytokinesis. This is not true, right? This is not what happens. Cytokinesis happens at the same time as telophase. Okay? So don't make that mistake. In interphase, we already talked about this stuff. Okay? The DNA is in chromatin. It's going to be replicated. It's in a clearly defined nucleus. The centrosomes and the other organelles that are going to help mitosis happen are being doubled up so that each cell has its own copy, and the cell is preparing to divide. We have this a little video here that we're going to watch. Hopefully it works. So here's the process. Interphase, we're making copies. Prophase, we're kind of preparing. Metaphase, you're going to line everything up in the middle. Anaphase, you pull them apart. Telophase, you create a new nucleus. And cytokinesis happens at the same time. It's hard to display in that video, right? But that's when you get the new cell. First off, prophase. This is the first phase, right? In prophase, what's happening is your DNA is going to start supercoiling. And remember, before it was in chromatin form, and after supercoiling, we're going to get our chromosomes. So this is really cool because it actually becomes visible under a microscope. And not today, but on Monday when we continue, we're going to be looking at microscope sides of this process, and you'll be able to see the difference between chromatin and chromosomes. There is a very distinct difference in how they look. So your chromosomes are going to be made up of genetically identical sister chromatins that are joined at the centromere, right? And I'm just going to change to a laser pointer here. Here's your sister chromatids, right? This is a single chromosome here. I'm trying not to highlight the other one. Single chromosome. It's created a genetically identical copy here. These are called sister chromatids, and they're joined right in the middle, pinched together at a centromere. These paired centromeres are going to start moving to opposite poles of the cell. Think of that as like the North Pole and the South Pole, right? Even though there's not really a north and a south in a cell, it kind of looks like, wow, what's going on? Stop it. Stop being weird. Okay, North Pole, South Pole, and it is erased again. Okay, there you go, it's stuck around. North Pole, South Pole. okay? These sister chromatids are going to be pulled to opposite sides during prophase. And they're going to form microtubule spindle fibers. Oh, sorry. These are your centromeres. They're going to, you can see they're on the north and south side. And you can see these right here. These are, I should have kind of maybe defined what I was talking about. These are your spindle fibers. I often refer to them, they're kind of like lassos, okay? So you have your centromeres, which are these things. You can kind of, I drew over top of them, but these are your centromeres. If you look closely, I'm going to zoom in. If you look closely, if you look closely, they kind of look like a nut in a bolt, right? This would be a bolt. They kind of look like bolts, and these centrosomes are going to create these wiry spindle fibers that connect to the centromeres of your sister chromatids, okay? And this is going to be kind of important. Think of it as like a cowboy lassoing a calf, right? And it's going to hook onto the calf, and then the cowboy is going to pull that calf in. In this case, your calf is the sister chromatids, and the lasso would be your spindle fibers, and the cowboy would be your microtubule, right? Who's lassoing in the spindle fibers. We're not going to be pulling the chromosomes, the sister chromatids, in yet. We're just lassoing them for now. Okay. And the last thing that kind of happens here is the nuclear membrane is going to break down. So you can see that here. I'm going to point at it. Here's you see the white dashed line. This used to be where your nucleus was, right? And we know that the nucleus has a nuclear membrane, a porous nuclear membrane around it. Well, in prophase, that nuclear membrane starts to break down and dissolve, okay? And so all that on the inside of your nucleus is normally all that chromatin. It's turned into chromosomes and then sister chromatids. And so now these sister chromatids are just kind of in the cell. They're not surrounded by a nuclear membrane. They're just loose in the cell, and that's when they're able to get caught by the lassos or the spindle fibers. Let's move on to metaphase. Hopefully this is kind of ringing a bell, right? You did learn about this in Science 9. In metaphase, the microtubule spindle fibers form from both centromeres, connect to the centromere of each chromosome. So here we go. They're lined up, well, you can see that they're lined up in the middle, and the spindle fibers are connecting to the centromeres, right? One on each end. Okay, so you have your sister chromatids, right? From this north end, the spindle fiber has connected to the north-facing sister chromatid. And from the south end centromere, the spindle fiber has connected to the south-facing sister chromatid. And you can kind of see what's going to happen next is they're going to be pulled apart, right? But that's not happening in metaphase. For now, we're just connecting. Next, the microtubule depolymerizes, and the spindle fibers start to shorten in length and contract. So here they're going to be starting the process of pulling apart. But that usually, once they start to get disconnected, we've entered anaphase. Here they're just starting to shorten, and it's not until they actually, they're going to shorten and tighten up, right? You can think of it as like shortening and tightening up, and once there's enough tension, they're going to pull apart. This causes the chromosomes to align along the center of the cell and the equatorial plane of the metaphase. So you can see what's happening here is right now these spindle fibers, some are long, some are short, right? In the metaphase process, what happens is these microtubules cause the spindle fibers to start to shorten in length and contract. And it's like if you had a bunch of rope and then you started to tighten it you end up getting to the point of least resistance where these microtubules are short, and with tension from both sides, they end up lining up in the middle until, like a tug of war, right? They end up lining up in the middle, and once there's enough tension being pulled as these spindle fibers keep getting shorter, they're going to pull apart those chromosomes. So let's move to the next step here. Anaphase. In anaphase, the spindle fibers keep contracting, they keep getting shorter, and this causes the sister chromatids to separate. So again, the spindle fibers kept getting shorter, that tension's building. Remember, they're in the middle, and eventually they rupture, they pull apart. Once the chromatids separate, they are each considered an individual chromosome. So before we call them sister chromatids, here they're sister chromatids, and here this is chromosome 1 and chromosome 2. And I don't know whether you can see the highlighter there, so I'm going to switch. Here, this is sister chromatids, and it has split into chromosome 1 and chromosome 2. But remember, these are genetically identical, right? They're exact copies of one another. And these genetically identical chromosomes move to opposite sides of the cell. So there isn't really a north end and a south end, but we're just using that for simplicity's sake. This chromosome 1 is going to move to the north end, and this chromosome 2 is going to move to the south end. Telophase. In the final phase, and remember, cytokinesis happens at the same time, once the two chromosomes arrive at their poles, so the north pole, south pole, those spindle fibers disappear. And you can see that here. So you can see that the spindle fibers are gone. The centromere is still here, but the spindle fibers have now fully dissolved. The chromosomes then decondense. So remember that in this form, this is a chromosome. This is highly condensed, highly structured, and you see here the chromosome has decondensed into its chromatin. And at this point, you wouldn't be able to see it under a microscope anymore. It would just look like a dark blob, like the nucleus looks. And the nuclear membrane starts to reform, okay? And we can kind of see that too. So here's your nuclear membrane. Again, it's here, right? It's starting to reform. Eventually, at the end of this process, you will have a fully formed nucleus with chromatin inside of it. And then finally, I've said this a few times now, cytokinesis is going to happen at the same time. And here you can see the cells are starting to split in half. You can see they're kind of rupturing in the middle in the cleavage furrow here. And we're getting to the end. Now we talk about cytokinesis, which is the actual splitting, right? It's when the cytoplasm divides and the cell membrane is going to be reformed around it, that fluid cell membrane. Okay, it happens differently in animal cells and plant cells. So we're just going to look at animal cells first. After anaphase, the microtubule filaments, they form a concentric ring around the center of the cell. So here you go. Here's the concentric ring around the center of the cell. The microfilaments are going to constrict to form a cleavage furrow. So it kind of, I would say, looks like a sphincter tightening, right? It's going to constrict, and when that happens, it's going to pinch the cells together. So you can see here, it's being pinched inwards, and this is called the cleavage furrow. When the furrow meets in the center, so when it pinches down enough and it meets in the center, the cell is going to become completely pinched off, and the two cells form. So here you can see it's pinched even further. Once it pinches completely, this is just going to separate, and this elastic fluid cell membrane is going to be pinched off, and you will have two cells instead of one. Okay. And because the separation happens from the outside and moves towards the center, it's called centripetal. There we go. Last slide of the day. What happens in plant cells? How is it different? After anaphase, so in animal cells it happened at the same time. After anaphase, you're going to have these rich carbohydrate-rich vesicles that are going to form a row at the center of the cell in what we call the equatorial plane. And you can kind of see that. Where's my laser pointer? You can kind of see that happening here. So remember that the cell wall of plants is made of cellulose, right? Which is a form of carbohydrate. It's fiber, right? We're going to get little vesicles packed with these carbohydrates, the cellulose, forming right in the middle of these two cells. The vesicles filled with the carbohydrates fuse together. So they become one large elongated, basically vesicle, right? And you can kind of see that happening here. These vesicles fuse into a long elongated thing. We call this the cell plate. So this is a cell plate here. And the cell plate is going to extend from one to the outward. It's going to start kind of in the middle, actually, not on the outsides. It starts forming in the middle and starts moving outwards until it connects to the two sides of the cell. And because it originates in the center and moves outwards, this is centrifugal. Okay. Thanks everybody for watching my video. If you liked it, please support it by hitting that like button. You can hit subscribe right here. I appreciate the support. And you can find future videos in the playlist right over here. And of course, the next one in the series, just click this link right here. See you next time. It's Mr. Emond. Bye-bye.